Any other statement about the enzyme?
No. Hello?
Yes, 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 I'm here. You don't know anything else?
No, I said no.
Okay, good. Now, the think about the enzymes. Before the enzymes, I would like you to explain to me about the catalyst. What's a catalyst?
They speed up the reactions without being used up.
Mhm. Speed up the reaction. Okay, now let me tell you about the definition, the true definition of catalyst. A catalyst is any chemical compound. Oh, okay, no problem. Any chemical compound when added when added into a chemical reaction reduces the requirements of that particular reaction Suppose, let's talk about it. You have a reaction going on A plus B, and you are making C from it. This reaction is requiring 100 degrees Celsius. This is the mode of the reaction or without any catalyst. But what did you do? You just added a catalyst into the same reaction, and the same reaction now is occurring at 45 degrees Celsius. What basically it is doing? It is reducing the demands of this particular reaction. Reducing the demands of this particular reaction, that is what we often call as catalyst.
Okay.
The maybe a reaction is supposed to be occurring at acidic pH. At in the acidic environment, the enzyme can make it to happen at a neutral environment or some other pH. So the catalyst, every catalyst have this particular quality, and that is they reduce the requirement. Now a question can be asked here, that why do different reactions even require temperature? I'm not talking about the catalyst. I'm talking about all the reactions that occur inside or outside the body. Why do they require temperature?
Because it increases the energy of the particles
Okay,
in the
it
reaction.
increases the energy of the particles, providing the energy. Okay. Any more particular statement?
No.
Mhm.
Do you mind sending this link again? The Jamboard, the link
Link
I have doesn't work.
this one.
Yeah.
Oh, sorry. Hang on. It would work. Hang on. It would work. Let me just change its access.
And then can you
Tell
send
me,
the
can you
link
access it?
again? Just in case.
Yeah, I can accept it.
Yeah, it's working now.
Important part is that the requirement of the temperature is because we need the particles to we don't need, basically nature needs the particles to collide. Whenever there is a reaction occurring, whenever there is a reaction occurring, the every reaction's particles must be colliding with each other so that uh, they can properly react to each other. Suppose there are two particles A and B, and you need them to perform a reaction, and they are at a very in a cellular language, they are at a very much longer distance to each other compared to each other. You need them to get closer. You provide the energy, you provide the temperature so that they can collide to each other and they can perform the reaction. What would happen? The catalyst basically basically makes the collection I'm sorry makes the collisions easier, and the reaction. Suppose suppose that previously uh, the temperature the temperature uh, required to collide the temperature required to collide the particles was 100 degrees Celsius. After adding catalyst, you know that according to the previous reaction that I drew over there, after adding the catalyst, they are now colliding at uh, 45 degrees Celsius. Another very important thing that we need to know about is the activation energy. It's basically connected. The amount of temperature, the required temperature to make the particles collide to each other, is called as activation energy. And basically, what a catalyst does, catalysts reduce the requirement of activation energy. Is it understood?
Yes.
They actually reduce the uh, requirement of activation energy. So the main thing about to understand about the catalyst is that they would always be reducing the activation energy or the requirements of a reaction. Suppose, okay, now the catalyst, if you try talking about the catalysts are of two types. One type of catalysts are called as industrial catalysts, and we basically we basically use them in our industries. We basically use them in our industries. You know that there are many industries in which the temperature we use is 2000 or 3000 degrees Celsius.
Yeah.
But if we add the catalyst in those chemical reactions, it becomes very easy for those reaction to occur. It becomes very easy. So that is why um, the industrial catalysts are necessary. But the same reactions, the same things do occur in the biological catalyst, in the biological system. So we name those chemicals as biological catalysts or enzymes. We name them as biological catalysts or enzymes. Now, as we know about the function of the catalyst, I don't think we need to know any more about the function of enzymes. How do they work and what happens? But a few pieces of information are necessary to understand about the functioning of enzymes, and that is when we talk about the enzymes are uh, also called as biologically active proteins. Now, this piece of information has two words that need explanation. What is the meaning of active? And the second thing is the enzymes are made of proteins. So basic chemical structure of enzymes is protein. What is the meaning of biologically active? Active substance is always the substance that basically active substance is always the substance that basically uh, directly participates in the reaction. Like if I talk about the function, the role of proteins in our body. The roles of proteins are three different. I think I have told you this. One is structural, they make our structure. Yes, I have told you this. The another role of the protein is functional, and the last role of proteins in our bodies are uh, metabolic. Metabolic. Now, the metabolically active proteins for the metabolism, we need metabolically active proteins. And okay, well, how would you define the metabolism?
The process of digestion, like how fast the food digests.
Metabolism basically is a bunch of reactions occurring all over in our body, all over in our body, not only about the digestion, but all over in our body. When they occur, we call them metabolism. And every protein that directly participates in a reaction, they actually directly participate in the reaction. That is why we call them what? Active. So remember that enzymes are biologically active. Enzymes are biologically active. So is it clear?
Yes.
The other, okay. And now the important thing is the other parts of the enzymes, the other things about the enzymes we need to discuss are basically the characteristics of enzymes. The characteristics of enzyme. But I also need to explain one more thing. How does a catalyst affect a reaction? Catalyst or enzyme? For that purpose, we have a graph in which we put the energy or activation energy on x-axis, on y-axis, and the time on x-axis. Suppose that the reaction is starting here, starting point. The reaction is starting here, and you have to provide energy, provide and provide and provide, and the reaction is happening here. Reaction is happening over here, and then the energy level goes down. Uh, the energy level goes down. Okay, so when the energy level goes down, it means the products have been formed. But remember that this reaction is um, non-enzyme catalyzed. Non-enzyme catalyzed. Okay, you would be looking at a word repeatedly. That word is called as catalysis or catalyzed. What is the meaning of these two words?
Catalyzed, so a reaction that already has a catalyst in it.
Yes, a reaction that is being controlled by a catalyst is called as a catalyzed. It is controlled by a catalyst, and it is called as catalyzed. But now this reaction, the graph that you are looking at, this graph basically is this particular graph is basically non-enzyme catalyzed, which means that this reaction is not um, catalyzed. But now you see another reaction in which you see the total activation energy that you needed to provide is this, the peak activation energy. And this reaction is catalyzed, meaning that the catalysis is involved. So when the catalyst is involved, then obviously the reaction's activation energy or required energy is moving down. This is the difference in the activation energy that you look at. So next is when the activation energy is reduced, the reaction can occur at a quicker pace. It can occur at a higher speed. So that is what is important in our bodies. Cleared?
Yes.
Now we are about to understand the characteristics of enzyme. The characteristics of enzymes are basically first of all, they are made of proteins. Made of proteins, and the proteins are basically spherical or globular in shape. Spherical or globular are almost similar terminologies. Spherical or globular means something that is in like a ball, a shape of a tennis ball. Another very important part is enzymes take part in a reaction. Take part in a reaction, perform catalysis, perform catalysis and uh, you can say come out of a reaction of the reaction without being used without being used or affecting the products. You know the rule that when you add another compound in a reaction, it ultimately affects the products. Is that
Yeah.
clear?
Yeah.
So basically, what do? You, when add the enzymes would just do the reaction. Enzyme would just do the reaction, create the product, and come out of a reaction as it is. Like A plus B plus X. X is the enzyme. And it would be converting A plus B into C and coming out as it is, ready for the next reaction. Another very important point is ready for the next reaction. So that is what the enzymes do. Okay, I'll number all the properties. Number 1, number 2. Number 3 is another very important thing. The reactants, the reactants or the chemicals being catalyzed by enzymes, being catalyzed by enzymes are called, what do we call those reactants or chemicals which are being catalyzed by enzymes? What do we call those chemicals?
Um, Reactants being catalyzed by enzymes.
The chemicals at which the enzyme act upon.
Sorry, I didn't get that part. There was some background noise and I didn't get that part.
The reactants or the chemicals being catalyzed by the enzymes. Or I can change the statement. The chemicals acted upon by the enzymes.
So is
Acted
it a
upon by the
substrate?
enzyme. What? Yes, acted upon. Those chemicals or reactants are called as substrates. So the reactants or chemicals being catalyzed by enzymes. Okay. So in this case, A and B are what? They are substrate. Another very important thing that we typically needs to know, that is important understanding of the reaction, understanding of the enzyme's activity. Number 4, not a whole enzyme structure is involved directly in a reaction. Instead, a small part, a small part performs the catalysis, performs catalysis called as active site. The remaining protein is called as globular structure. Globular structure or supportive structure. Globular or supportive. Okay, what is the meaning of this whole sentence? Okay, when we sit on the chair, do we use the whole chair? Do we sit
No,
on the whole chair? No,
no, you-
we don't sit on the whole chair. We sit on a particular part of the whole chair. It means that in our direct use, we only use a small part. But if you use any other part, like the hand rests or the legs or the back of a chair, the whole structure of the chair would be compromised, obviously. It would be compromised. So similarly, a small part of the enzyme that actually involves in a reaction. This is the ball or the globule of the enzyme. This is the globule of the enzyme, and the shaded region is the globular structure. This is the globular structure. This whole is the enzyme. This small part is it directly involves in a reaction, and it is called as active site. It is called as active site. The remaining part is what I have told you, called as globular structure. And it is a reality that if globular structure globular structure is destroyed, the active site would be distorted, would be distorted, and the catalysis won't occur. So, which means that both have equal importance. Both have equal importance. But yes, the active site needs to be supported so that it can perform a reaction. So that is why active site and globular structures, these are really, really important in the enzymatic reactions. Clear?
yes,
Okay. Now, about the next point that for the characteristics of enzymes, about the next point, we would have several subpoints for it. Basically, in this particular reaction, in this particular point, we would be discussing about the mechanism of enzyme action. Mechanism of enzyme action. First of all, what we need to know about it as a point A, the active site and substrate have to be complementary to each other. Hold on, don't write this. Don't write this. I first need to explain another point. Forgot that. When we talk about the mechanism of an enzyme action, the uh, the enzyme and substrate first have to bind to each other. Bind to each other. Otherwise, no reaction would occur. Would occur. Like, if I properly write it, how do we often write normal chemical reactions? A plus B goes to form C. This is just a random chemical reaction, right?
Yes.
But when we write the reaction with the enzymes, substrate plus enzyme, first the substrate and enzyme have to bind to each other. They have to bind firmly to each other, which is called as ES complex. They have to bind. And then ultimately the substrate would be converted into product and enzyme would be coming out of it. It is just like it is just like let me give you whole when you create have you do you know about like when we create the different kinds of utensils, we need the cast. We need the cast for them.
Yeah.
You know about? We need a cast for them. If you want to convert the glass, or maybe you are making the utensils with the soil, which are also called as matti ke bartan. Have you ever seen that?
Yep.
What happens whenever you want to make them perfectly? You would be putting them into a cast so that they would be getting the shape of that cast. They would be getting the shape of that cast. If you want to make a proper utensil, you have to first make sure that a cast and the material should come in contact properly. Cast and material should come in contact properly, and then later you would be separating the cast from the finalized utensil or the finalized structure. Overall, by all this, we mean that whenever we are trying to convert a substrate into a product, first the substrate have to come in contact and bind with the enzyme so that it can be converted into the product. So this is basically the mechanism of enzyme action, that enzymes don't randomly perform a reaction. They have to first form the ES complex. Remember, if it is not formed, no reaction would occur. A very clear statement. ES complex has this much particular importance. Fine.
Yeah.
Another very important point that we'll discuss now: how an ES complex forms. Okay, for this purpose, we need to understand that the active site and enzyme and substrate have to be complementary to each other, have to be complementary to each other. What is the meaning of complementary?
That they help each other, or like they come together.
Like they are
They
exact
work together.
opposites to each other. They
Hmm?
are exact opposites to each other. They fit in.
Okay.
They fit in. So that is a complementary. You can say they work together. So to make a perfect ES complex, you need to have the shape of the substrate as same as the active site. As same as the active site. That is what the requirement demands. Remember, this word is used quite often when we talk about the enzyme and when we write our answers in the CIEs. This word is the key word. They have to be complementary to each other. And remember, so that they can form an ES complex. Sorry, I forgot to complete it. Like this. Enzyme, substrate, this is ES. Another thing, the active site, due to complementarity is so much is so much uh, specific that it cannot fit any other substrate, just like a lock and a key. Just lock and a key. You cannot fit any random key into a lock, right?
Yeah.
Just like that, you cannot adjust the any other substrate into this active site. The active site has to be complementary, just like a lock and key. The lock and key model basically refers to the specificity of enzyme. Enzymes are extremely specific and they have to be adjusted. Just like in this model, the enzymes act as a lock and the substrate act as a key. So you can simply say that suppose this is a lock or enzyme and there are two keys or substrates. One is like this and the other one is like this. Obviously, you know which one would fit and which one would not. So remember that enzymes are specific due to which they don't even attach to any other substrate. They are specific. Okay, this was the third point about the enzymatic action. And another very important point about the enzymatic action is enzymes. Uh, okay, yeah, enzymes need to collide, to their substrate to complete a reaction. I have a small question, and the question is, we say that enzymes work best at a particular temperature. Have you
Yes, I've heard of that.
ever heard about? Yeah. So it means that, why do you think that enzymes would require the temperature?
I don't really know why they would need a specific temperature.
No, no, no. They would need a specific temperature, but why? What did you say? What was your answer?
I don't know about why it needs it.
See, uh, there is no doubt, there is no doubt that enzymes do reduce the activation energy or demand of activation energy. But there is a there is a very serious fact that enzymes do need to collide. And for collision, the energy is still required. Energy is still required, which means that whenever the enzymes are reacting, whenever the enzymes are reacting or catalyzing a reaction, they still need the temperature to collide and contact with their substrate so that they can complete a reaction. That is why they require the temperature. Clear?
Yes.
Okay, next. So, this was about the mechanism of enzyme action, and this is how the enzymes basically contact with their substrate and how do they complete a reaction. Yes, complex formation is really important. Okay, now the thing is that uh, we are about to discuss the. Let me see if I have left something. I'm just trying to get over with the doubt. Okay, we are almost completed. Now, um, let's talk about how many points have we discussed till 5 points. Now, we are going to talk about the sixth point. The sixth point. Um, Rate of enzyme action. Enzyme action. Okay, what is the rate of a reaction?
How fast a reaction takes place, or how long it needs,
What
how much concentration is like that.
what would be your standard to detect that how fast a reaction is taking place?
By measuring the concentration over time
Concentration of what?
of your end product.
Yes, basically we uh, measure the given amount. Oh, sorry, the formation of a product. Formation of product in a given time. That is called as the rate of reaction. Like enzymes, basically, they can speed up a reaction. How? They can speed up a reaction just like there is an enzyme. There is an enzyme that can convert 100,000 molecules of substrate molecules into products in 1 second. 100,000 molecules of substrate into products in 1 second, just in 1 second. That's quite impressive. So, when these enzymes work, then is there any factor? Is there any factor that could affect the rate of action? Affect the rate of enzyme action? Do you know?
No, I don't know.
Okay. Before understanding it, um, okay, let me tell you two types of factors. One types of factors are those which uh, interfere interfere with enzyme enzymes physical features, and the other types of factors are those that interfere with enzymes performance. Now, in the second category, the topic comes in A levels. But for the first category, we have two factors that we need to discuss. One is the temperature, and the other is the pH. Okay, before understanding all this concept, remember that the enzymes are enzymes are globular proteins, as we have already spoken about it. And the globular proteins, uh, you must know that the globular proteins are made of multiple, like two or three smaller proteins, smaller proteins bound together, bound together by strong, bound together by strong, not strong, by some supportive bonds. Supportive bonds. Those bonds, I'll tell you the names of two bonds: hydrogen bonds and ionic bonds. Suppose that this is an enzyme. This is one protein. This is second protein. This is a third protein. This is a complete enzyme. And all of these proteins are uh, being bonded together by the supportive bonds. The problem is that the supportive bonds of enzymes, the supportive bonds are really sensitive to temperature and pH changes. If they break, if they break, all small protein subunits would separate. Enzyme would be enzyme or protein would be broken, would be broken, properly called as denatured. Okay, now let's see. The Like, have you ever boiled an egg by yourself?
Yes.
Hmm. If you don't boil the egg, just break it. What are the colors you can see?
Transparent and yellow.
Transparent and yellow. Good. If you if you boil the egg, then break it, what color can you see?
White and yellow.
Hmm. Now, where did that white come from when there was no white before boiling it?
So the it became the egg whites before the transparent ones became white.
The egg white, after boiling, became white. Otherwise, it was transparent. Is that what you were saying?
Yeah.
What happened to that egg white that suddenly appeared different and a bit solidified?
A reaction took place.
No. Basically, By boiling, you have denatured the protein. By boiling, you have just denatured the protein. And once you denature the protein, it changes the shape permanently. You can write on your notes there that once the protein is denatured, it changes its shape permanently, thereby thereby no that enzyme is of no more use now. That enzyme is of no more use now. So remember that every spherical protein would be denatured when you boil it, no matter it is enzyme or not. Every globular protein would be denatured. So now what you are saying, if you are looking at enzymes, if there are enzymes and you are giving them a very high temperature, basically their supportive bonds are breaking. And when they are breaking, they are ultimately denatured. So the word denatured I have written in the inverted commas. It is as important a word as I told you about the complementary. The two words, the total crux of this whole chapter of enzyme is two words. Enzymes are complementary, enzymes would be denatured. All the rest of it is just the supportive concept. Believe me, most of the questions in the CIEs are related. They just ask almost the same thing. They are complementary, they denature. They are complementary, they denature. The rest of the concept is supportive. Why would they denature? Like obviously, I have told you why would they denature. Okay, Another problem would happen with the denaturation. Denaturation would lead to denaturation would lead to lead to a distorted structure of active site. Now no more complementary to the substrate, so no ES complex would form. No ES complex would now form. Simply putting it, no ES complex would be created, and ultimately it would be just broken. It would be just useless now. Cleared?
Yeah.
So now, how do the temperature and pH affect, or how do they act? Temperature. We don't explain these things much. We just create the graphs that would explain themselves. If you give a vast variety of temperatures to an enzyme, over here you put the rate of reaction, and there you put the temperature. 0, 10, 20, 30, 37, 40, 50. Now, when you would see the rate of reaction after increasing the temperature would suddenly rise, and it would reach at the peak up to 37. Then it would remain a bit stable, and suddenly it would fall when it would move above 40. Now, first of all, we need to understand that over here the enzymes are what? Denatured. But before it, what is happening before it? Before it, the collision between enzyme and substrate are gradually rising, gradually rising, hence increasing the rate of reaction. And at this temperature, the optimum collisions are happening. Optimum collisions are happening. So this temperature would be called as optimum temperature. And that is a 37 degrees Celsius. And you must know that uh, what I was about to say. Yes, from 0 to 37, with every 10-degree rise, 10-degree rise, the rate of reaction increases twofold. What is the meaning of twofold?
That doubles.
Doubles. With every 10-degree rise, it would be doubled because the collisions are increasing. And ultimately, it would be denatured. And like not all biological uh, objects, I should say living things require same uh, optimum temperature. Like if you see, most of the time the animal enzymes, I'm not talking about the human, animal enzymes mostly require 40 degrees Celsius as optimum temperature. Some bacteria, plants also require, mostly I'm saying, mostly 40. But and the bacteria mostly have the same demand, but they also have a optimum temperature of around 75 degrees to 85 degrees. Around 75 degrees to 85 degrees, and they just like obviously they are very stable enzymes. Why it happens? There are many bacteria that live at um, many bacteria. They live in such hot springs, the water of which is almost 80 degrees Celsius or 90 degrees Celsius. There are literal hot springs in a very high altitude area of Pakistan, Siachen. In Siachen, there are some areas, some you can say some particular springs in which the Siachen is basically a disputed territory, and there are the army is always present over there. So the soldiers there, they put the eggs in that hot spring and they just boil the eggs there. They just boil the eggs over there. Now you can imagine that what kind of temperature would be there, and literally the bacteria live in those at those temperature. How do they live there? They have thermostatically stable enzymes. Thermostable enzymes. So that is why it is very obvious that they can tolerate that optimum temperature. Got it?
Yes.
Let's talk about the pH. When you do, how the pH actually affects. What is actually the pH?
The acidity level or the acidity level.
The acidity level or the alkalinity level, or in other words, you say the basic level
Yeah.
of the solution. So we often say that the 1 to 6.9 is acidic, 7 is neutral, 7.1 to 14.0 is basic.
Alkaline.
Yes, alkaline. So enzymes, the pH affects in two ways. Number one, it can change the fitting, it can change the charges on enzyme and substrate, on enzyme and substrate necessary to form ES complex, to form ES complex. Suppose this is an enzyme and it has these charges minus, 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 plus, 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 and this is a substrate having plus, 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 minus, minus, minus. Now these opposite charges are necessary for them to join and to complete the reaction. The pH would be affecting these charges. If these charges are being affected, no ES complex would form. And the second effect of the pH is the acidity
Mm.
or alkalinity can interfere with the the acidity or alkalinity can interfere with the supportive bonds. And you know what would happen when they would be interfered?
What
Enzyme
happens?
would be denatured.
Denatured.
Yes. Enzyme, when the supportive bonds are interfered, the supportive bonds are broken, they would be ultimately leading to the denaturation. For this purpose, you can create a graph. Over here, you have rate of reaction. Here you have the 0, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. There are many enzymes you would see that which are showing the very high activity at 2 and their activity finishes at 4. Their optimum pH is 2. Some enzymes would start their activity at 5, but their optimum activity is 7 and their activity would fall until 9. Some enzymes are there, they would start their activity at 7, but their highest activity is shown at 9. Until 11, they are denatured. Like I can tell you, the enzymes, this enzyme is pepsin present in stomach. This enzyme is amylase present in saliva. This is the pH of saliva. And this enzyme is any enzyme present in the intestine. There are multiple. So the pH can vary. The demand of pH can vary according to the. There are some enzymes which work best at 4. I won't tell you about those, but they work best at 4. And all these pHs exist at in our own bodies. Got that?
Yes.
You know what? Your chapter is finished.
Okay.
Now, I would just like you to be very quick and have a quick look at the whole lesson.
Okay. 1 second.